First of all, I have to say that in PPILOW, our task was improving the robustness of laying hens and piglets against parasitic and bacterial infections, which we took over along with Aarhus University, who took care of the chickens, and our part was the pigs. So as you saw in the poster of Sana and Ricarda, they presented already their results. Unfortunately, they couldn't be here today. This is just to give you an idea about some of the publications which led us to the partnership in this project, and some of them which resulted from the project, which I'm going to present further. Just as a brief introduction to the entire work we've carried out so far and which is ongoing in PPILOW, was the fact that the parasitic diseases have a considerable effect on pig production, causing economic loss due to low immunity, morbidity, and mortality. There is a huge problem nowadays in the entire world with chemoprotection, treatments against parasites and against bacteria. Everybody knows about antimicrobial resistance, right? And plants, phytotherapy itself, could represent a valid, biologically available and cost-effective alternative to this kind of classical therapies. The use of local remedies, plant remedies, has increased notably in the last decades due to their biodegradable traits, decreased toxicity if used correctly, obviously, environment friendliness, and to some extent, as far as we know from literature, anti-parasitic, antimicrobial, and immune-stimulating effect. We had three main directions of study. One was to use plants against parasites, see their effects. The other one was to use plants to control the bacterium of the pigs which are raised on the outside. In low input, free range systems. And the third one was, why not, to combine this with immune stimulating effects of the plants. If we could achieve all three by using different plants or plant combinations, that would be wonderful. Mostly because we try to use, as you see in these pictures, plants which are locally available, very common, like garlic or pumpkin, or calendula, or summer savory, which everybody grows in their gardens. Furthermore, some of these plants are already in use in our traditional raising system of pigs, which I have to tell you, just to avoid any confusion, that it's most commonly a mixed system. So what we confront in Romania is, well, farmers they are called, but it's not exactly a farm. When a family has 1 cow, 1 horse, 3 pigs, 2 sheep, 20 chickens in the same barn. So to avoid the confusion and lack of control in such a system, which is 90% of the pig industry in Romania, it's more for family consumption. We did our experiments, which you will further see in a so-called, you heard yesterday about a so-called, I'm not sure, as my colleague talking yesterday wasn't sure, conventional outdoor system. So we found a farmer who had several pigs and then another one, and they had this, well, not access to the pasture, broad field, but these pens on the outside where the pigs had access. So the initial steps were to see where we are. So look at the prevalence of swine gastrointestinal parasites in 2 free-range farms from northwest region of Romania. And I already explained the aim, because there are lots of problems caused by parasites, health problems and welfare problems leading to economic loss. This part of the study envisaged a segment which aimed at identifying the prevalence of the parasites in 3 age categories: piglets, fatteners, and sows. The materials and methods were those we use normally to control parasitic infestation. I'm not going to go into detail. The references were images which are part of a very broad database. According to use of this database, we identified several types of parasites, including protozoa, but also including nematodes, as you see. These results indicated, and I'm not going to go into figures, details, the three authors of this presentation and many more behind who did the work. So the three of us are here, so you can ask further questions if you are interested in punctual details. I just wanted to underline here on the overall prevalence, that there are differences between weaners, fatteners, and sows, and the distribution of these parasites is different by age category. We found different parasites in higher percentages in all categories, or we found parasites in very low percentages like Strongyloides ransomi in just a few of the categories. So the point here is that there is a difference between the distribution of different parasite species by age category. We also looked at the seasonal differences in these parasites, and as you can see in this is the sows, this was the weaners and fatteners. So you can see the differences by season, blue being spring, summer, autumn, and winter. Apparently those parasites which were more prevalent persisted during at least one season. So as a conclusion of this part of the study, it provided essential information to start out with in Transylvania's distribution of gastrointestinal parasites in pigs. The parasites were present in most pigs reared in free-range farms in the study area, so both farms indicated that the parasites were present, which is not a very encouraging information. This information is not only available and useful for our further study, but also is useful for farmers, policymakers, and researchers, leading to potentially better pork production for public consumption. And then, of course, the control strategies need to be implemented. But before that, we need to raise the awareness among pig farmers, as I told you, because these results can be extrapolated to these small households which have pigs. The other part of the study, we moved on to looking at the effects of plants, namely garlic, Artemisia absinthium, pumpkin, coriandrum, summer savory, and calendula marigold, on the embryogenesis of Ascaris sum eggs during an in vitro experimental study. So we did the epidemiology, then in vitro studies, and then we moved to in vivo studies further. Ascaris sum is one of the parasites which is present in traditionally managed farms, and you can see some images here. The problem of increasing anti-helminthic resistance led to seeking alternatives, maybe some traditionally used already, medicinal plants being included. And this study aimed at evaluating the in vitro effects of the above-mentioned plants on the egg hatching and larval development of Ascaris sum. This is just a reminder of the life cycle, and very important is to remember that the larval stages have a migration in the internal organs of the pig, which leads to multiple damage, and now a very shocking image, I would say. Look at the parasites in the intestine, right? It's loaded, and look at the lesions, necrotic foci in the liver of such a pig, where the larvae migrated. and then a more shocking image leading to the idea that in fact these are zoonotic parasites. So they can be really, really very dangerous for people. And when we talk about these small households we have, it's even more important to raise the awareness about the risks and try and use all the means we have to control such infections. We looked into before starting to administer the plants even in vitro, we looked into their potential to treat such infections, to destroy the eggs or adult stages of the Ascaris and that's why we looked at the chemical composition of the plants by using HPLC and gas chromatography, mass spectrometry, to identify the active compounds. Of course, we had a protocol. The eggs were collected from pigs, randomly collected from pigs raised in traditional system, and then, of course, you have the protocol we worked with. We used all the 6 plants as alcoholic extracts, and we looked at the hatching percentage, and here you have the stages: an embryonated egg, the morule, early morule, the egg with first larval stage, and then the 2/3 larval stage, and this is the hatched egg with the larvae next to it. What the results indicated for the concentration in different active substances of the plants, everybody knows that polyphenols are very active both against parasites and bacteria. I'm not going to talk a lot about that. Just to have an idea that these plants, which we selected based on their presence in the pastoral households where these pigs were raised, contain have very different content, just to correlate that with further with the biological effects of different active substances, isoquercetin here, for example, or other components in different plants. There are different components in higher quantities. So these plants actually they are different from each other. They are not ranked by their content in one certain component. Here you see the analysis of plant extract activity, and you have presented here the reactivity of the plants compared to the alcohol control and distilled water control. So this is the distilled water, the blue. You see how low it is, both at 5% and 2.5% concentrations, and how the plants inhibited the hatching. Quite high percentages for all the tested plants. And here you have lower concentrations, 125, 0.625, and 0.312. These were the 5 concentrations which were tested as their activity for the inhibition of hatching of the larvae. All plants, as I said before, showed various degrees of inhibitory effects on egg development. Better, or the strongest anti-helminthic effect, were shown by Allium sativum, that is the garlic, and then Artemisia absinthium, like ranking them, the pumpkin and the summer savory. Then, based on these results, we could suggest some of these plants could be used in combinations as new anti-parasitic herbal remedies. This was one of the first, or to our knowledge, the first study of ethnopharmacological properties of these plants used to treat Ascaris infection in Romania. Then we moved to look at, because the parasites were not only worms, but roundworms, but were also protozoa. So we looked at their efficacy, these plants' efficacy on protozoa. The in vitro anticoccidial activity of some herbal extracts against Amyris species. We didn't differentiate between different Amyris species. Why? Because the coccidia are present in pigs, but their clinical effects are very rarely expressed. It has to be conjunction, a complex of different favoring factors in order to have the clinical disease. Nevertheless, it could be there, so if the animals are suppressed, it could be there. Here you see some clinical aspects of the coccidial diarrhea in piglets, very young piglets, but here you see the hemorrhagic lesions in the intestine due to congestion of the blood vessels due to aimeriosis. In order to diagnose the disease, it's useful to look at mixed feces samples. And here you see the Isospora suis oocysts. So what we've done here, it's a common procedure in microbiology, looking at the minimal bactericidal, minimal oocidal concentration, minimal inhibiting concentration. So my colleague and student here today, Mihai, applied this method. And the results obtained were again compared to the standards, looking at unsporulated, sporulated, and destroyed oocysts by microscopy, subsequently to exposure to these concentrations of plant extracts. We used alcoholic plant extracts all the way in vitro, right? So these were including the chemical composition, including the anti-helminthic properties and anti-protozoal properties, all were alcoholic extracts. And here you can see very well the decrease of the sporulated oocysts and the increase by all plants, there are different plants, and the control, increase of destroyed oocysts due to the effect of plants. And you can see that the maximal values at 72 and 96 hours were obtained in summer savory extract Saturea hortensis. All samples were tested in quintuplicate, so 5 times repeated, so we could process statistically the data. And here you see the table with the quintuplicate replication of the experimental study. This was one of the few studies performed on Eimeria oocysts isolated from piglets. And the statistical analysis showed that plant extracts were effective both in inhibiting sporulation and of oocysts and destroying these. Just a minor percentage resisting to this activity of the plants. Out of the plant extracts tested, the lowest LC50 values were obtained in Calendula officinalis, marigold, Artemisia absinthium, and Coriandrum sativum. These were the most potent. Our in vitro results demonstrated both inhibitory and destructive effect of the plants, so eventually they could be used as disinfectants in those shelters where oocysts are present. The implementation of herbal formulas which contain the most effective alcoholic plant extract is needed to obtain the stronger anticoccidial effect. And we aimed at further investigation on isolation, purification, toxicity studies, and mechanism of action of the mentioned plants and their compounds. In vivo studies, as I told you, we moved from in vitro to in vivo studies in parasites. One of them envisaged the assessment of antiparasitic effects of Allium sativum and Artemisia absinthium against gastrointestinal parasites. You'll see that we divided the plants in groups of 2, but giving them separately to each group, target group of or age group of the studies, right? So Coriandrum sativum and Cucurbita pepo, and then in the last experiment in vivo, Saturea hortensis and Calendula officinalis were tested in parallel. For the first study, we aim to evaluate the in vivo effect of all plants, as I said, and we divided plant by plant on 2 low input farms in the Transylvania area. The methods were the same. These results were actually already published in 5 publications so far, and some are on their way, so this is why there is a repetition of the methods, as you saw probably in posters in Perugia. There is the protocol, the dosage protocol which we used in 10 weaners, 10 fatteners, and 10 sows. The calculation of the plant, you see there is a difference between the amounts. We tried to calculate based on their weight, on the animal's weight, obviously, and tried to use the maximal non-toxic concentration of the plants for each age category. Here you see some of the results of chemical testing of the plants, which we already have been through. The results indicated again the presence of multiple parasites while looking at the parasitic load before the treatment. And here we can see the percentage of fecal egg oocyst count reduction in these two plants tested. The two colors indicate the two farms. And this experiment, probably I didn't mention in the beginning, we used two farms which had two different breeds which are considered local breeds in Romania. One was Mangalița, everybody knows about, and the other one was the black pig, which has in the scapular area white stripe. We've seen that as under an Italian name breed and Spanish breed. So the breed seems to have or to be local in several areas of the world. Both plants we tested in this experiment had administered as food supplement for 10 consecutive days, had a strong antiprotozoal and antihelminthic activity, Allium sativum being more active than Artemisia absinthium. So both have the potential of treating gastrointestinal parasitosis in swine. And the antiparasitic efficacy can be attributed to the presence of multiple chemical compounds such as polyphenols, tocopherols, flavonoids, sesquiterpene, lactones, and sulfoxide. The next test involved, as I told you before, two other plants of the 6 we selected, Coriandrum and Cucurbita. And the protocol was exactly the same, and we compared the two farms in terms of the efficacy against parasites of these main isolated parasites of these two plants. And based on this study, we concluded that both plants' powders at the dosage mentioned before for 10 consecutive days per oral administration were efficient. Coriander was more effective against protozoa, while pumpkin showed efficacy more against helminths, the nematodes we isolated. Considering all the constraints of Romanian livestock farming, these results are a beacon of hope for better management and welfare practices in swine farming. In addition to the best of our knowledge, this is the first ethnopharmacological report on the antiparasitic effects of Cucurbita pepo and Coriandrum sativum, traditionally used in Romania for treating protozoa and nematode infections in swine. The last study compared Calendula officinalis and Satureia hortensis, marigold, and summer savory, and here you have the composition of the two. Again, we used the standards to compare the cysts and eggs that were isolated, and here you see some of those. The results indicated differences on two farms between the two plants which were tested against parasites, with higher values in Calendula officinalis, for example, against Eimeria on one of the farms, but lower on the other farm. And then obviously higher results in nematodes also were observed. In conclusion, again, both plants showed promising in vivo antiparasitic activity, while Calendula had a stronger antiprotozoal activity and mildly antihelminthic effects. Satureia hortensis was very effective against both helminths and protozoa infections. The antiparasitic efficacy can again be attributed to different components. Maybe it's worth mentioning that we didn't use separate components. We used the entire plant. Why? Because this is how it is found on the pasture by the animals on one hand, and this is how some of the owners administer these plants. The extraction would be very, very costly. And then even if some of the components could be administered separately with obtaining better effects, this is again at risk because the owners might not want to invest and might not want to strictly control the dosage administered to the animals. And some of the compounds could be toxic at higher levels. So as I said so far, the plants we tested were active in vitro, active in vivo, and this is why we moved a little bit further, because it's known that parasites carry a lot of bacteria around, and during the larval stage of migration or just by their presence in the intestine, they damage the balance of the microbiota, intestinal microbiota. So we looked at the potential of alcoholic plant extracts to control pathobionts of swine on a low input farm. This research aimed at looking at the antibacterial properties in vitro of these plant extracts. What we've done was just classical microbiology, collecting samples, oral samples from the pigs. We inseminated those samples, isolated and identified the bacteria, and we further also monitored the antimicrobial resistance of these bacteria. And surprisingly, we found very high antimicrobial resistance in some individuals on some of the farms. Meanwhile, we did the aromatogram placing along with, in parallel with the antibiotics on other plates, in the wells, the alcoholic extracts of the plants in different dosages. These are the extracts I was talking about, and this is an aromatogram, another aromatogram example. We isolated very different, these are the plants again, very different bacterial population, and you have here the description, the name of the species and the strains we isolated. This seems really very crowded, but these are the inhibition diameters obtained by checking the antimicrobial resistance. And these are the aromatogram results, and here we see, we use the 6 plants mentioned already. We see that the highest average of inhibition was encountered in Coriandrum sativum, and the minimal was for the pumpkin. So there is a difference between, if you compare the studies so far with these studies, there is a difference between the antiparasitic and antimicrobial activities, not the same for each plant, right? So this is why they have to be tested before use. What is really worrying us by this result is that the MAR index, and urges us to try and obtain better solutions based on these plant extracts, is that the MAR index, I don't know how many of you know what it is. It's the division of the antibiotic numbers to which the bacterial strain was resistant over total numbers of antibiotics used. So if I use 10 and the bacteria was resistant to 8, that it's 8 divided by 10, so it's 0.8, right? And from 0.2, the bacterial strain is considered to be resistant. So this 34, in a system which doesn't really use antibiotics, not because they would not be needed, but they are expensive and the owner cannot afford, is really worrisome. So in conclusion of the bacteriology, we can say that some of the tested plant extracts could display considerable antimicrobial activity on pathobionts. These plants could enhance the welfare of the animals by reducing the potentially pathogenic antibiotic-resistant bacterial load as an alternative to classical antibiotic therapy. Further, we expanded a little bit this research, and we decided that E. coli could be one of the pathogenic bacteria in swine, which is really impacting on human health, consumer health, and also the animal welfare, because there are some diseases, like 5, caused by E. coli in very young piglets, in weaned piglets, then in fatteners as well, and sometimes mastitis in sows. So we expanded this research, and we found that these plant extracts were efficient against antibiotic-resistant E. coli. We had a poster in Perugia, so we have that data as well. And the last segment of our research was the investigation of immune potential. This time we did some research in vitro, again using the plants and looking at the development of mononuclear cells, which are lymphocytes and monocytes in whole blood cultures, additive with these plant extracts. And based on those results, we decided to look at the in vivo, in parallel with the in vivo tests during the administration of Artemisia absinthium. This is a segment of the study, and we looked at the changes in the growing capacity, multiplication capacity of the mononuclear cells. So the point in this study was that immune suppressive circumstances are very important in defining the health and welfare of the animals. We were seeking, meanwhile, to have plant origin or plant-based immune stimulating, immune modulating products in order to potentiate the host to increase its ability to control infection, diminish the synthetic drug consumption, and prevent antimicrobial resistance. Also in this study, we aim to test the tolerance to oral administration of Artemisia absinthium, testing the in vitro spontaneous and mitogen-induced cell-mediated immune responsiveness, and testing the in vitro effects of other plants in the animals which were already treated with Artemisia. We, of course, tested the extracts, as I said before. The dosages of Artemisia absinthium were established based on the data in literature. The research was carried out on Mangalitsa farm, extensively raised pigs, on suckling piglets, weaned piglets, and sows, 10 for each group, as you've seen in the previous studies. The feed was supplemented with 5%, 5 per mil Artemisia absinthium L granulated, which was administered in the daily ratio for 7 to 10 days per group. Blood was sampled before the treatment, at the end of the oral treatment. The blood was cultivated in the specific culture medium, and then it was divided in 200 microliter aliquots and was added with 1.5 microliter per well of all the extracts I already mentioned. Here you see the cultures after 48 hours of incubation in a 5% carbon dioxide atmosphere. We measured the glucose consumption by a specific method. So here you see the composition of Artemisia we started with. The extract of Artemisia had an antibacterial, antimicrobial property. It has been considered to be this extract a promising medicinal component or additive to therapy. Based on its chemical composition. As I said before, it's anti-parasitic and it's anti-protozoal. Some data of the literature which says that vulgarine has a strong effect against multi-drug resistant bacteria, and something about beta-cytosterol, which we found in our extracts in our plant. Here you see the results. In suckling piglets, the cultures had a stimulation index, which was the highest for marigold, right, the threshold. And then after, it's interesting, it changed that the pumpkin showed the highest effect in stimulating the growth of immune cells. So somehow the Artemisia administration sensitized the piglets to the active compounds of the pumpkin towards the end of the treatment. Here you see the results for the weaners, where the pumpkin was the threshold, and you see some negative results. That is possible because of the hemolysis during the administration of Coriandrum sativum. Again, here we had just a few dosages, and looking more into different dosages could solve our problem with this negative indicator here. In sows, actually you see that the results are quite even, and none of the extracts showed, of course, the graphs have the values here on the side, so you can compare the different categories. We also had a minus here, but none of the extracts showed really stimulating results in this age category. So Artemisia absinthium actually had a negative effect on the specific immune response when administered orally in pigs, suggesting the eventual reconsideration of the administration dosage and protocol. That was our conclusion. So it was anti-parasitic, anti-coccidial, antibacterial, but still it did act negatively on the immune system. And thank you for your attention. That was all.
Thank you, Marina. We have 5 minutes
Questions?
for the discussion. Yeah.
I just want to understand very well, what is the final object? Is it to produce new drugs, or to give advice to farmers to use this plant or the other one? Because
And more.
okay, so because I think farmers use mostly water extract, and in your study you use
Alcoholic extract. We used alcoholic extract because this is mostly used in experimental trials. So we wanted to compare our results, which were not carried out in Romania, but some were carried out elsewhere, to see if we are on the right path. And as I told you, it's a very big research, and actually the plants are administered, for example, pumpkin, they just chop it and give it as it is. So you eventually, because it is watery, could consider it's a watery compound, so on. But in watery extracts, the active substances are very mildly extracted, so you won't have those many, right? So the idea was to prepare something which can be added to the feed they administer to their pigs, not necessarily the plants as they are, but we had to start somewhere, right? And then prepare something, obviously, when we have enough data, and compare all the data, select on a rigorous dosage, because we just had some doses here, as you saw, prepare something from the most active, best plants, which are anti-parasitic, antibacterial, and immune stimulating. So far, garlic is on that path, but we still, because it has some toxic compounds, we still need to be careful. So yes, it's raising awareness against what could be. You've seen that dramatic picture of a very sick child. In piglets is the same. They could be also very sick, right? So raise the awareness. Provide them, just implant the idea that there are some natural means to control this. It doesn't have to be very expensive, right? Because they are like, oh, it's expensive, we can't do it. And then they drop it completely, right? And they will not, the good thing is they don't really buy drugs, because it's as it is, and then because drugs are expensive. And unless the vet says that this needs something like that or that. And this is why the antimicrobial resistance is really scary, because it's quite high. And actually we found some of this antimicrobial resistance higher in these small households than on intensive raising of pigs. Why? Because they have the biosecurity there, and they tend not to use drugs. They limit the access, they limit whatever they can to avoid introducing those pathogenic bacteria. While here it's free range. So they go to the pasture, they go with cows, they go with sheep, they go with geese and ducks and whatever creature you can imagine, wildlife on that same pasture during the night. So, and this is uncontrollable. So they need to know what they are facing. That's one. And the other one is trying to prepare something which is affordable because it's local resource. It's scientifically sound because we tested it and we found out that this specific plant is best. And then try to agree with some producer who can also do the technology for not too much money, you know. Is it satisfactory? Okay.
Yeah, sorry, just very quick one, out of curiosity, do you have any records of self-medication actively...
Of what?
Self-medication actively displayed by the animals roaming on pasture?
Very few because of the observation. The observation is very difficult when the household is handled by the family. Today somebody releases the pigs, the next day it's somebody else, and they will not, even if the pig is sick, let's say, they will eventually keep it in the barn, so there is no plant self-medication on the pasture. And they try to do it in the barn, so it's not self-medication. And then if the animals are healthy, then every day somebody else will take care, and the observation is really very difficult, as Cesare mentioned before, under these circumstances.
Parasite is a main fundamental topic when we act about extensive systems. So, and we are trying to develop some different approaches from intensive system and low input system. So we know that a little burden of parasite is not harmful for the animals at all. So regarding the first part of your presentation, and that is absolutely interesting. But I, as a farmer, a farmer as I am, I will be interested more in respect to prevalence, more like a quantitative analysis that could be more understand not the presence of the species that is so important for zoonosis. You are perfectly right, but to understand the fluctuation of season and also because we are working with local breeds. So also the threshold that we could read on the leaflet of many signs of the parasitic drugs are suited for intensive system animals that are more important than the ones that we use. So I think we have to also foster this research and to underline new thresholds for animals locally based, and that could be also tested when animals have some decrease in health status, decrease in welfare state, and decrease in production as well. So I think it's a great, there are some also tools for on-farm quantitative analysis of a program, so something that makes farmers more independent in the action of any usage for self-medication or something natural with respect to the other, but when is necessary, because also parasites can be trained by the constant use of some natural substances, so when they are really needed, they cannot react as they could if we use when it's so necessary because the burden is so high. So thank you very much to open finally this topic that I think is one of the most challenging. It's enormous. Thank you.